Hello, and welcome to my presentation of genetically altered animals. My name is Owen Shriver, and today we're going to be exploring the world of genetically modified animals. Now, to begin, genetically modified animals are usually given an additional trait for their DNA. This is usually for better survival or just experimental purposes. My project we will be looking at the chipmunk. Now, the new trait we'll be giving it will hopefully let the chipmunk change color depending on its environment. Now, I would like to get started. To get started, we are going to look at what animal and gene we are going to be using for it. For this, we're going to be using an ordinary male chipmunk. The gene that we'll be giving this chipmunk is called chromophore. This is a very special trait because it comes from chameleons. Chromophore is a gene that uses cell signaling. These signals can be hormones that are initiated by changes in mood, stress, or local environment. In this case, it will depend on local environment. We will hopefully take this gene from a chameleon and insert it into the DNA of a chipmunk. Now, I'm going to get a little bit more scientific. As you can see here, we have two different pieces of DNA. The gray being the chameleon, and the blue being the chipmunk. We are going to split the DNA in half using restrictive enzymes. The enzymes act as a splitter for the DNA because they're able to split open the DNA, making us able to insert the chromophore from the chameleon into the DNA of a chipmunk. As you can see there's two pieces and they become one, the gray becoming one piece with the blue. This is hopefully what happens, and the chipmunk and chameleon gene can become one. But you're probably thinking to yourself, how is this useful for chipmunks in their environment? Well, I have the answer. Chipmunks are small rodents, usually primary consumers, on their own food chain. There are many different animals like hawks, snakes, and even humans that would like to kill them for meat or for any other reason. Chipmunks usually stand out in their environment, like you can see on the pictures on the left and right, but we're hopefully able to fix that, making them not easy to find and eat. With this new gene introduced into chipmunk, we can go from sticking out, like the picture on the left, and being blended into the environment, like the picture on the right. This will hopefully occur, and chipmunks will be able to change their fur so they cannot be caught by prey. But the only bad thing is that there are some down effects of this. Even though the chipmunk is difficult to find, which sounds like a good thing because they will live longer, it makes them very rarely eaten. So these chipmunks may over-reproduce and become over-repopulated, and the consumers previously of these chipmunks may suffer themselves because they have nothing to eat because of the chipmunks disappearing. Now, before we perform this experiment, we should probably know about the gene that we're going to use. The gene is called chromophore, and it's found in all chameleons, which makes them so special. With the chromophore, it uses cell signaling, which results in a color change, making them very hard to find. This is called background adaptation. The cells are able to mimic the background, making it seem like the animal is a part of the background. This is what we hope to accomplish with the chipmunk in their own environments. Now, we are going to look at the process of actually performing the experiment. So how can we do this? The steps to genetically modify this animal is we first have to find and isolate the gene wanting to be used. The gene can be isolated with enzymes. Now, once we isolate the gene, we can remove the gene and split open the DNA of the chipmunks using restrictive enzymes like previously. Once we place in the gene or the chromophore it will hopefully go into the chipmunk DNA and become one with the chipmunk DNA, making the chipmunk have this trait and be able to function well with the new trait. So you can see the picture on the right, this is what we hope to happen. Take the yellow piece of DNA from the chipmunk and then place it into, from the chameleon, and place it into the chipmunk. Now, with this gene in the chipmunk, we can finally complete our genetically modified animal. The chipmunk will have fur that is able to change color depending on its environment. This will be the first time it has ever been done. But to fully understand and make sure these new animals function properly, we're going to make sure that they reproduce. Now, as you can see here, we have a Punnett square. The top being a non-GM animal and the left being a genetically modified animal. So, all their babies will be carriers but not actually be able to use the trait. Eventually, there will be more and more carriers, and they will eventually have the trait. So, we are able to observe if this will actually work. Now, let's just recap what we know. First, we learned that we'll be using chipmunk and chromophore from a chameleon, and we're hoping that the chromophore from the chameleon will help the chipmunk change colors. With this trait, they will be hidden from prey and not be eaten. And to do this, we will isolate the chromophore, take it out from the chameleon, and insert it into the split DNA of a chipmunk. Then, hopefully, the genetically modified chipmunk will produce, populate, and will grow in that environment. Thank you, and I hope you have enjoyed my presentation.